Okay, so we're going to start with active transport. And we know passive transport is going down a gradient. So active transport is going up a gradient or against a gradient. So moving molecules up a gradient. This is often called moving against a gradient. Using energy from ATP. Okay, and it doesn't always use energy from ATP, but we're just going to say, for the purpose of this discussion, that it does. Okay, we'll talk about some instances today where maybe it doesn't, but yeah. All right. So we're going to look at a specific instance of this. It's called the sodium-potassium pump. Okay, and the way that the sodium-potassium pump works is that it is a enzyme that can change its shape based on whether or not it has a phosphate group attached to it. And you can imagine where the phosphate group is going to come from. It's going to come from adenosine triphosphate, right? So whether or not it's got its ATP added to it or not is going to determine its shape, okay? So it's going to look something like this, and obviously it doesn't look anything like this in real life, but this is a good way to sort of show its function in a model. Okay, so here's what it looks like in one of its states. And the other way that it looks like, we'll say, the other thing that it can look like is this, where now it's going to point out. But now the funky things, the squiggly lines, are on this part. Now these are little triangle notches. Again, it doesn't really look like this, but it's useful in visualizing it. Starting over. All right, so this one on the right is what it looks like when it's been phosphorylated. So we're going to add a phosphate group in here. Like that. And we can highlight it or something to show that that's where the energy is. Okay? All right. So, in this example, this is going to be on the outside of the cell. And this is going to be on the inside of the cell. And the way that you get from one shape to the other shape is you phosphorylate it. Phosphorylation. Or you add ATP. That's what phosphorylation means. Okay, ready for this? We've got our drawing done. Now we can talk some actual science. All right, we've got on both sides we're going to have 3 Na ions. These are Na plus ions. We're going to draw them in circles. And then on the other side, or on both sides rather, we're going to have 3 K plus ions. And I'm going to draw those in triangles. I'll grab that so you guys can finish drawing. Okay, so we're ready to see how this works? All right, so you can see here, this is opened up to the inside, and it's got the little circular active sites here in this enzyme. And so these sodium ions are going to move into those active sites. Okay, so then they're in those active sites, and then it gets phosphorylated, right? And when it gets phosphorylated, now we've got these 3 sodium ions that can't fit into those spots anymore because they've changed their shape, and so they then go onto the outside. Okay, so now we're going to end up with 3 more sodium ions than we started with on the outside. And then when the potassium ions join, when the potassium ions go into these spots, it causes the phosphate group to break off. And when the phosphate group breaks off, it turns back into the other type of, or the other side of this diagram here, where it's going to then dump the K plus ions back into the inside of the cell. Okay? So, the function of this, in a nutshell, is to move Na plus ions out of the cell and allow K plus ions in a ratio of 3 Na plus to 2 K plus. Okay? So we can say it's called an Na plus K plus ATP ACE, which is usually just called the sodium potassium pump. And we can say that its job is to it moves Na plus ions out of the cell and K plus ions into the cell in a ratio of 3 to 2. Questions on this so far? All right, so this is the big new thing that we're going to talk about today. The other thing that we're going to talk about today, we said we're going to do active transport, passive transport, and cell signaling. I want to give you guys a real example of how these things function and how they all coordinate to allow a cell to do its job. Okay? So, that's what we're going to do down here. And Steven, that was Wade. He wants to see you. All right. So, we're going to look at a neuron. Okay? A neuron is the main cell of the nervous system. And you've probably seen one being drawn before. It looks something like this, where you've got a cell body that's sort of circular like this. Okay? Then you've got something that's called the axon. This. And the axon goes to these things called the axon terminal. something like that. Okay, and then on this side of the cell body, you've got these things called dendrites. Like so. Okay, so again, this is the axon, this part of it. This is the axon terminal. This is the cell body. Sometimes it's called the soma, depending on how old the text is that you're reading. Okay, and then down here, these things are called dendrites, these little offshoots. Okay, that's not really all that important that you memorize those for this class, you know, later on maybe. Okay, so we're going to look at how all these things are going to be involved in allowing the neuron to function. Okay, so all along the axon, there are going to be a couple things. There's going to be sodium-potassium pumps, which I'm going to denote as something that looks like this. That's an Na plus K plus pump. Then there are something that looks just like straight up and down like that. That's going to be K plus channel protein. And then they're going to be a box, and that's going to be an Na plus gated ion channel. Okay, and they're all going to line this space here where you've got a bunch of the sodium-potassium pumps. This, you got a bunch of K plus channel proteins, and then you got a bunch of gated ion channels. Okay, so the other things that we're going to put in here, we're going to have the red dots. Those are going to be Na plus ions. The green dots, or if you don't have those, you can make triangles, I suppose. We'll do triangles. Those will be K plus ions. Okay, and the sodium-potassium pump, like we said, is going to move sodium ions out and it's going to move potassium ions in. The way that I always remember this, that my psych teacher always taught me to remember it, is that the neurons are salty bananas, right? Because they have potassium in them like a banana does, and they're salty like you put salt on them because the salt's on the outside, right? So a salty banana is a neuron because it's got potassium on the outside and sodium, or potassium on the inside rather, and sodium on the outside. That's how I always remember. I mean, it sounds ridiculous, but I never forget it, so it must work. All right, so what we're going to do is show these sodium ions on the outside. So this sodium-potassium pumps are pumping the sodium ions out, right? So these things are getting pumped out. And we're going to end up with a bunch of sodium ions on the outside. And a few of them on the inside, but not many because they're being pumped out. Lots of sodium ions on the outside, like so. Okay? Now we got to talk about potassium. So potassium is being pumped in, right? So we're going to end up with more potassium on the inside than we have on the outside. There's a few on the outside, but mostly they're on the inside. Okay, so let's talk about diffusion. These sodium ions, where are they going to want to go? Where are there more sodium ions? Yeah, there's more on the outside, so they're going to want to go in, right? Can they go in? No, they're positively charged, and therefore they cannot go through the plasma membrane. They're stuck out there, okay? The potassium ions, where are they? They're inside, so where are they going to want to go? Outside. Can they?
No. Yes.
Yes, why? Because there's a potassium channel protein, right? So the potassium channel protein allows for these potassium ions to duck out of here, right? They're going to be able to go through these channel proteins. Okay, so not only are we pumping more positive ions out than positive ions in, because we're doing 3 sodiums out and only 2 potassiums in, but then the potassiums can also escape back to the outside. So which side is more positive, the outside or the inside? The outside is a lot more positive, okay? In fact, we say that the resting potential of a neuron is approximately negative 70 millivolts on the inside. That means that the difference in electrical potential on the inside and the outside is around negative 70 millivolts. Okay? Let's write some stuff down that we've done. Okay, so the Na plus K plus pump what it does is it moves 3 Na plus ions out and 2 K plus ions in. And the result of that is that you increase your voltage difference between the outside and the inside, making the outside more positive and the inside more negative. So the outside becomes negative. Or sorry, the outside becomes positive rather, and the inside becomes negative. That's because we're pumping 3 positives out and only pumping 2 positives in. Right? The next thing that happens is that the K plus ion channel protein allows K plus ions to escape, to go to the outside. Allows K plus ions to diffuse out of the neuron. And this makes the outside even more positive and the inside more negative. Now, everybody in here should have heard at one point in their life that a neuron, or that our nervous system, uses electrical impulses, right? That's how our nerves send messages to our brain, is using electrical impulses, right? Well, if you've ever wondered where the electrical impulses come from, this is the way that we create electrical potential, is by pumping ions out of the cell in order to create a positive ion on the outside and a negative on the inside, right? Okay, so we still haven't talked about how this thing actually works then. So the last thing are these gated ion channels. So the Na plus gated ion channels. Okay, we can say that these gated ion channels are only going to open when the differential in voltage reaches negative 55 or less. Okay, so when the voltage difference is negative 55 millivolts or less, these channels open and allow Na plus ions to diffuse in. All right, so here's the magic. Let's say this is a sensory neuron, okay? And so me touching stuff causes it to be stimulated, all right? So there are proteins that are in the dendrites here, and they are stimulated by touch, okay? When they get bumped, they are going to be stimulated. These are signaling proteins. Okay? So when they get bumped, the thing that happens is those things are going to release Ca2 plus ions into the cell. Okay? Into the cell body. So they get bumped, they get stimulated, and they release these Ca2 plus ions. That's calcium. Okay? Well, you're releasing Ca2 plus ions into the cell body there. What happens to this magic number? This negative 70 millivolts? What? It goes up. It goes closer to zero, right? Because it's not as polarized now. Because you're adding positive charges to the inside. Well, if enough Ca2 plus ions get into the cell, it might reach that negative 55 millivolts. If it reaches negative 55 millivolts, what happens? The gates open, right? And if the gates open, that means that all these NAs that are out here are going to rush in. They're going to go in so fast. They're going to fly through these gates. And that's going to depolarize or cause this end to get more positive, which is going to open the next set of gates, which opens the next set of gates, which opens the next set of gates, until the inside of the neuron is now extremely positively charged, which causes some signaling proteins on the other side that are going to respond to positive charge within the neuron to release their neurotransmitters out into the synapse, okay, which is the space in between neurons. Okay? If you relied on active transport or some sort of chemical messenger to do this activation here, it would take forever for a signal to get from your foot to your head. Okay? But we're not relying on that. We are relying on the diffusion of an electrochemical gradient. Right? So it's sending this charge down the neuron, right, by opening up these gated ion channels. When it gets to the end, it signals these proteins to release whatever their neurotransmitter is, and that's going to trigger the next neuron, which sends a pulse down that axon, which triggers the next neuron, which sends a pulse down that axon, and that's how a neuron actually functions. Okay? So in order to get a neuron to function, a lot of things have to happen. Some of the stuff is passive transport, some of the stuff is active transport. So let's talk about the stuff that's active transport. There are two active transport events in the firing of a neuron. What's the first one? What has to happen via active transport? It does require energy. Yeah, it's going to use energy in the form of ATP. But what thing are we powering with ATP here? Yeah. Sodium potassium pump. So Na plus K plus pumps are powered with ATP. Okay, and there's one other spot. It has to do with those Ca2 plus ions. So the Ca2 plus ions are pumped into signaling proteins using energy from the diffusion of Na plus ions. So in other words, the sodium potassium pumps are working really hard to get all these sodium ions out, right? But there are parts of the cell in the cell body, parts of the neuron in the cell body that are going to allow Na plus ions to come in, and they're going to use that energy of the Na plus ions coming in to pump the Ca2 plus ions into the spot where they need to go in the signaling protein. Okay, so those are two different types of active transport. One of them uses ATP. One of them uses the motive force of Na plus ions traveling through or traveling down their concentration gradient. Okay, passive transport. Which things were passive transport? Yep. K plus ions, yep. Moving into, or sorry, out of the cell. Via facilitated diffusion. One other thing. Where else was passive transport happening?